Hi there. Last time I had talked about some things about what cells are. So we went through this exercise where I talked about the sizes of each one, the relative sizes, and then blew a cell up a million times and then shrank it down a million times at the end. And then I talked a bit about the relative cell sizes of bacterial cells and some eukaryotic cells and how those compare with virus sizes as well. Then I briefly discussed the parts of a bacterial cell, then parts of a eukaryotic animal cell, and parts of a plant cell. And these are all general pictures, right? And then went through also a few facts about sizes, some numbers of things, and all in an attempt to get you to think at the cellular and molecular level, get down to those levels about thinking about some of these things. Then we briefly discussed the tree of life, looking at these separations and when approximately bacteria separated from archaea, and then we got the first eukaryotes. And diversification of all of these as well, and that's one thing I want to talk about today. And part of this diversification was first diversification of unicellular cells, and those are all the ones in white here, so including the bacteria, archaea, and then all of these up here are eukaryotes. And then going into the diversification of cells within multicellular organisms as well. Then I gave an internet definition of a prokaryote, but said that it's really not that simple, and the term prokaryote is not a good way to define cells, and that's because it pretty much is a good definition of these bacterial cells, that they are cells enclosed by a membrane, and they have an unenclosed DNA, their chromosomes. But the definition does not adequately describe this type of bacterium, and this is one type of a planktomycete, which does have internal membranes and has a structure, a double membrane, that looks like a nuclear envelope that surrounds the DNA, the chromosome. So, and there are other planktomycetes that have other internal membrane-bound organelles, and so it really doesn't fit that definition. And even then, the term prokaryote means before or without a nucleus, and normally we don't define things by what they are missing or what they don't have. We define them by what they have. So in this class, as I said, we'll use the term prokaryote only briefly once in a while, and instead I'll deal with bacteria and archaea specifically. Then I described a few archaea. Many of them are extremophiles, but many of them are not don't live in extreme environments. So originally they were all considered extremophiles, and now we have examples of mesophiles and things that are not extremophiles. Then I went through briefly a bit of what a eukaryote is, and eventually described eukaryotes as organisms that are very good at developing endosymbiotic relationships with other organisms, and so they have multiple organisms inside of other organisms, and so that's and also they have these internal membranes, which they probably got from planktomycetes. So a lot of special characters for eukaryotes. And finished up describing this organism, Giardia lamblia, which only has nuclei, and it has two nuclei containing the same DNA, but it's a double nucleate organism, but has no mitochondria. So this is one that's a very simple organism, and it is parasitic, so if you happen to drink it in your water, you're likely to get a disease, mainly severe diarrhea from these guys. So now I want to start talking about cell diversity, primarily in eukaryotes, and I'll start down here with a group called the Excavata. And this group does include Giardia lamblia, so these are some of the earliest branching eukaryotes, so you would think they'd be very simple. But then there are some, like Tracheomonas and Euglena, that are photosynthetic. But their photosynthesis doesn't come from a chloroplast. This group picked up, by an endosymbiotic event, a eukaryotic alga, which was already photosynthetic, that had a chloroplast inside of it that came from a cyanobacterium. But in this case, it's a eukaryote inside of a eukaryote, and so that's how they gained their photosynthetic ability. This group of organisms also includes trypanosomes, which are not photosynthetic, but they do have the original cyanobacterial and algal genes that were incorporated during that endosymbiotic event, picking up the eukaryotic alga. So what's happened here is they've lost their ability to photosynthesize by losing some of those genes that allowed them to be photosynthetic, and instead they've adopted a parasitic lifestyle. So because they are getting many of their nutrients from the parasite, they don't need to photosynthesize anymore, and so those genes pretty much fizzle out. Or a better way of putting that is those genes mutate essentially out of existence. So the next group of organisms among the protists, the protozoans, are the Hecrobians, a pretty interesting little group. And this is because they build little shells around themselves, so protective shells to avoid predation. And again, many of these are photosynthetic because their ancestors also picked up or incorporated a eukaryotic alga within their cells, an endosymbiont that became the organelle. And there's quite a bit of diversity, as you can see here, as far as the elaboration of those shells that they build around themselves. And I always have to remind myself that these are single cells. These are not multicellular organisms. That's a single cell. Each one of these is a single cell with an elaborate set of plates that it protects itself with. And the next group up here are the Rhizarians, or Rhizaria simply. So these are quite diverse also, and again, these are individual cells shown in each case. So they surround themselves also with shells made up of a variety of compounds. Most are photosynthetic. So some of you know these as the foraminifera, and you can find these often on beaches, and you have to look in a microscope to see them, but some of those pieces of sand that you see are made up of these tiny little cells. And in most cases, within this group, they've also picked up a eukaryotic photosynthetic member, endosymbiont, that has become the organelle. But in one group within the Rhizaria, they have picked up a separate cyanobacterium, similar to a chloroplast, but they picked it up independently. So there have been at least 2 cases in evolution where a cyanobacterium has become an endosymbiont of one of these protist cells to become a photosynthetic organelle. In the case of plants, it's a chloroplast, and in the case of one in this group called Pollinella, it's known as a chromatophore, but it's analogous to a chloroplast. So the next group up here is the Alveolata. It used to be included as the Chrome alveolata, But those have been split originally in the into the Chromista and Alveolata, and now into the Stramenopiles and Alveolata. And it's a very important group. And again, within this group, many are photosynthetic. And again, they picked up their photosynthetic ability through an endosymbiotic event between a eukaryotic alga, a plant essentially, and a protist cell that was not originally photosynthetic. This includes dinoflagellates and some other groups. And again, each one of these is a single cell, so they are quite elaborate. Often the dinoflagellates have two flagella, one that emanates from the pole, and then one that kind of straddles the midsection of that cell, so they can move in 3 dimensions quite easily. So if you've ever heard about red tides, those are caused primarily by dinoflagellates, where there are these blooms of specific species of dinoflagellates, which also infect the shellfish and the fish, and actually will kill the fish. And then if you eat the shellfish that have ingested the dinoflagellates or become infected with the dinoflagellates, you can die also, because they carry a toxin that is a neurotoxin, and there's no cure for it. So if you eat enough of that toxin, it essentially shuts down your muscles and your breathing and your heart, and you pass away. And that's known as paralytic shellfish poisoning. And actually, I had a friend from high school who died from that. He went to a reception for a wedding where they were serving crabs, and this was up in Alaska, and noticed a tingling in his lips when he ate this crab. And by that evening, he had died of this paralytic shellfish poisoning, because some of the crabs served at the wedding reception had been infected with these dinoflagellates. The next group is the Stramenopiles, and within this group, again, one of their ancestors picked up a photosynthetic alga as an endosymbiont, which is now allowing them to become photosynthetic. So that organelle originates from a eukaryote. There are some members, however, that have lost their photosynthetic ability, and so they need to live off other organisms in order to get their carbon compounds. So this group contains this very large group of diatoms, which form silicaceous walls, cell walls, to protect themselves, and most of these are also photosynthetic. It also includes brown algae, and if you've ever seen kelp, giant kelp, those are brown algae, and they're called algae although they're not plants. They're actually protists, but they're multicellular protists within this group of Stramenopiles. It also includes these oomycetes, and these were once thought to be fungi, but they're not fungi, they're Stramenopiles, so they gain some of their functions from fungi, and so that's why they have some similarity to fungi. But these are also known as water molds, and so they infect fish, they infect plants, they're in the water, of course. And actually a wide variety of these. One group infects potatoes and soybeans, and so the potato famine in Ireland was caused by Phytophthora infestans, which will infect the potatoes. And you can see it in some of your potatoes when you get them. They have these kind of black areas within them, and those are usually caused by this Phytophthora. They also, another species, Phytophthora soji, infects soybeans. And so you'll see fields around here where there's been a lot of puddling of water in certain areas, and you'll see that the soybean plants are dying in those areas, and that's caused by this oomycete, which is not photosynthetic, by the way. It's a parasite that grows on plants and then eventually, kills them by primarily by just destroying tissue within those plants. There are many others, some that aren't pathogens, and they're just saprobes, meaning they eat dead material. But then there's another one that affects fish farms called Saprolignia, and you can find it out in Lake Erie also, which infects fish and actually will disable their gills, so they pretty much suffocate to death. And the oomycete, in this case, the Saprolignia, will live on the fish cells, the tissues from the fish. So within all these protist groups that I've talked about so far, many have their ancestors picked up a eukaryotic endosymbiont that became the photosynthetic organelle. Some have lost that ability to photosynthesize. And then there's the one group in Rhizaria that have picked up a cyanobacterium, a bacteria, rather than a eukaryotic alga. And those have become the chromatophore organelles in those, rather than a chloroplast in plant cells. So plant cells have chloroplasts, these ones have chromatophores, but they're very much analogous, although their origins are different. They were picked up by those protist cells independently in two independent events. So next is the Archaeplastida, or Plantae, the plant phylum. And these include unicellular algae as well as multicellular algae, all the way up to higher plants. And it was some of these unicellular algae that became the organelles in the Excavata, Hacrobians, Rhizaria, Alveolata, and Stramenophiles. So the three main groups that started out in the Archaeplastida were very early on red algae and glycophytes, and then later on green algae. But in each case you have unicellular types of red algae, and you have multicellular versions of red algae. And you can see sometimes red algae don't quite look red, but they still have the same types of chloroplasts within them. And the chloroplasts in this case did come from a cyanobacterial ancestor, an origin of cyanobacteria. So in this case, it is a bacterium leading to the organelle in these eukaryotes. Also, among the green algae, you have unicellular species, and you have multicellular or aggregate species, and then true multicellular species as well. And in some of the multicellular species, most of the cells look pretty much the same, whereas in other species you can have a diversity of different cell types in those multicellular organisms. And you can see this more readily in higher plants or vascular plants, in that they have different types of cells, both in the leaves and the shoot apices and the root apices. So here in the leaf you see there are palisade cells on the top that contain chloroplasts. There are spongy cells down below, mainly for gas exchange. There are stomata that allow gas exchange into that leaf, and then there's vascular system here. In the shoot apex, there are special cells at the very tip that control where a leaf is formed and how many leaves are formed in that vegetative or floral apex. And then in the root apex also you find certain cells that give rise to all of the other cells of the root tip, including these cells at the end that are the leading edge, essentially, of that root tip. And those cells at the end protect the root, the inner part of the root, from being damaged by going through rocks and soil and things like that. So in the unicellular organisms, we were talking about cell diversity based on species, the diversification. Now we're looking at diversification of cells within an individual plant or an individual organism, so that you have a diversity of cell types within one individual. Then we get to this next group, this branch that includes amoebae, fungi, and animals. And the amoeba up here, fungi and animals, none of them are photosynthetic, so they only have mitochondria and they have no photosynthetic organelles. The amoebae group contain quite a few unicellular members and also some multicellular members. If you know slime molds are within this group, and those are almost always multicellular, having a number of different cell groups or cell types. And in this case, with these organisms, they have no cell walls, so they spread out and move around, just as you would think an animal cell might move around, but it's a completely separate group from animals. Sometimes you can see slime molds on different surfaces, especially when gardeners are spreading bark mulch and things like that in the spring, and they'll appear as these kind of cream-colored patches. And if you pass them one time of the day and then another time of the day, you'll see that they are actually moving. And so those cells are moving as a group and eventually will form a reproductive structure. And in that case, it's not just one individual. Many times it's more than one individual coming together for the purpose of reproduction, but then they are cooperating, joining together to form the reproductive structures. And here is another major group, the fungi, which are joined together in what's called the Opisthokonta, because they do have similarities. So animals and fungi are related back in the past as one large group, but the fungi split off, becoming fairly diverse. And one group of fungi is called the Dicaria. And within the Dicaria, all of their cells, at one point at least, are not diploid at all. They're dikaryotic. So they have two nuclei coming from one each from the two mating types of that group. And they only fuse right before meiosis. So you can have haploid cells, you can have dikaryotic cells, and you can have, for a very brief time, diploid cells just before meiosis to produce more haploid cells. And we'll talk in more detail about those later in this semester. So here are a couple examples. N means haploid. That's the number of chromosomes. N+1, N is in the dikaryotic stage. So here you have in Phylophora, these are all haploid cells, including the haploid spores in a basidium, which just underwent meiosis in the cap of the mushroom. So this happens in the cap of the mushroom. Then they put out 4 haploid cells, haploid spores from each one of those basidia, which just underwent meiosis to form those 4 haploid nuclei. So if you look at a mushroom, right, most of the mushroom tissue, if you look at it very carefully, is this. It's dikaryotic, and they have these specific bulges between 2 cells when they divide. So there's N+N throughout that whole mushroom, except in the gills. In the gills, you would find these basidial cells, and those are the ones where the 2 nuclei finally get together to form a diploid cell briefly, or a diploid nucleus, undergo meiosis to form 4 haploid nuclei, 4 haploid cells, which become the 4 haploid spores. So quite a complex way to go through reproduction in the fungi. And over here, this is the reproductive structure in an ascomycete, and this is called the ascus. So in the basidiomycetes, you would have a basidium. In the ascomycetes, you would have an ascus. And in each case, they are the product of meiosis, forming the haploid spores there. And down here, this is Aspergillus nidulans. Some Aspergillus species cause disease, so you breathe them in and get aspergillosis, which forms these little black hyphae within your lungs and can give you a pulmonary disease. But these are all haploid. So you have haploid cells, and then you have haploid spores being formed by these reproductive structures also. So a lot of elaboration in ways to reproduce in the fungi, and a lot of different cell types then, cell diversity within the group and within each species. And then here's the other major group within the Opisthokonta, and that's the animals, Animalia. And while most species within the Animalia are multicellular, there are a few unicellular members of this, and the one major one are the choanoflagellates that look quite a bit like a sperm, and actually physically and their physiology, they look a lot like a sperm cell within this animal group. They do coagulate and communicate and also form sometimes what look like multicellular aggregations, but they're really individuals coming together for those purposes. So this is the group that is way down at the base of that leaf on the tree for animals, one of the original members, the original species of animals. And then branching out from that, here's the choanoflagellates. You have first the porifera, which are the sponges. So they have several cell types and control of those cell types. And then the placozoans, a little more complex. The ctenophorans here, which are like sort of like jellyfish, but not quite like jellyfish, a completely different group. And then the cnidaria, which are the jellyfish and allied groups, and finally the Bilateria, which includes the vertebrates and other larger, more complex organisms. So you build up different cell types as you go further and further into this group, including neurons, which are only in the Animalia. And by the time you get to very large mammals, including Homo sapiens, us, then you have many, many different cell types, over 200 different cell types in each one of you, including liver cells, and there are many different types of liver cells performing different functions. Then there are lymphocytes, many different types of lymphocytes in your bloodstream. And then even among muscle cells, there are skeletal muscle cells and there are cardiac muscle cells, and many of the muscle cells are multinucleate, and so you have other types of elaborations of cells. Then you have skin cells also. Many skin cells are not just diploid, but they're tetraploid because they're getting ready for cell division in case you cut yourselves. So those cells are ready to patch up any cuts, any break in the skin, almost immediately after the cut is made in the skin. So here's some other cell types. Here's an oocyte with a sperm ready to fertilize that cell. There's a neuron, one type of neuron, and there are many types of neurons. Red blood corpuscles, your red blood corpuscles have no nucleus. If you look back into things like dinosaurs and birds and reptiles, their red blood cells, they actually have red blood cells that still have a nucleus. So there is some variation there in the diversity of different cell types. And here's a lymphocyte compared to red blood corpuscles. You can see the large nucleus because it's expressing certain products that it's putting out on the cell surface to communicate in the immune response. And there's a T cell with HIV, with the human immunodeficiency virus attached to it. So not only do you have cell diversity, but you have other things attacking those cells. In this case, a virus. You have bacteria attacking those cells. Also, you have bacteria helping those cells. So in your body, you have, in and on your body, you have as many bacterial cells as you have your own human cells. And those cells are doing things like protecting your skin, helping you to digest your food, providing you with amino acids, many other things. So cells don't really exist separate from one another. They're always communicating with one another, interacting with one another, sometimes attacking one another. So there's a diversity of unicellular organisms. There's a diversity of cells within multicellular organisms, and there are also diversities of cells interacting with other cells. So that's the main focus of this video today. So just a few other cell types. Here are mouth epithelial cells, and you can take a toothpick and actually scrape a few off and look in a microscope and see these quite easily. Here's the cancer cells. Cancer cells often round up as if they're ready to divide. Normal cells will round up only when dividing. Cancer cells sometimes lose their cell structure and become round, looking more like an embryonic cell ready to divide. Here are some pancreas cells. And again, as you probably already know, there are many types of cells in your pancreas. Some help to digest certain types of food, and then others produce insulin so that sugars can move from your blood into your cells. Here's an endothelial cell, also showing the cytoskeleton, which is composed of microtubules, actins, and myosins, and also intermediate filaments. To stretch that cell, to stretch an arm of that cell or a piece of that cell so it is going to move in a certain direction, or to lose all that structure to become a cell rounded up, ready to divide, or, as in the case of cancer, losing that structure because of the difference in the biochemistry now of a cancer cell. And then finally a bone cell down here. So just think about the diversity of all these cells, both unicellular in the case of most bacteria and archaea, and most protists, as well as multicellular versions of those, increasing the cell diversity and cell-to-cell communication. And we'll talk more about cell-to-cell communication later in the semester also. And so this week in lab also, you'll be looking in microscopes at different cell types, just to look for yourself at the cell diversity that's out there. And there's a lot of cell diversity out there. So that's it for today, and thank you very much.